Hey there, AP, pre-AP bio students. We're ready to start on the microscope lab. So I've got my compound microscope lab set up right here. I've already have it plugged in. There's the plug. So a couple things I want to address to make sure you guys understand is we've got the eyepiece up at the top right here, and it does have a magnification of 10 times. But then we also have, these are the objective lenses that you see right here, and there are a couple of these. The two that we use for pre-AP bio, this one you can see the number 10. It magnifies 10 times the original size. So if I'm using that, that's low power, then I take the eyepiece as 10x and I multiply it times this low power 10x, and my total magnification is 100 times. If I'm viewing something on high power, and you can see right here, our high power is 40, then my total magnification is 400x. So I'm gonna start off on low power. Okay, that's the 10x. I'm gonna click it into place. There are two knobs that we're gonna use. This is the coarse adjustment knob, and I want you to look when I turn this, the stage, which is right here, is going to move significantly. Okay, you can see it move just like that. So the coarse adjustment knob moves the stage a lot. The fine tune adjustment knob, this one, is just gonna do little bitty increments. So the key is when I'm on low power 10x, I can use both knobs, but I'm really gonna use the coarse adjustment knob. Okay, that's gonna be the big one that I use. When I'm on high power, I never, ever, ever wanna touch the coarse adjustment knob, because if I start cranking this while I have it clicked in, you can see how close the stage is to the objective. I can crack that lens. So when I'm clicked into the 40, the high power, I'm only ever gonna use this fine-tune adjustment knob right here. Okay, so that's the microscope. Okay, so we're gonna start the lab. This is the lab sheet. I copied mine in yellow and I put them in sheet protectors to keep them safe. You guys at home have a set of these directions with you at home. You should also have an answer sheet by your side to use along with this. So the first thing I want to zoom in on is we're going to be constructing, and you can see it's this part right here, we're going to be constructing some scientific drawings. So as you do your drawings, there are a couple of rules you need to pay attention to. Number one, you have to use a pencil on all your drawings. Number two, you're going to color them, but you're going to use colored pencils, matte pencils. Do not use markers or Crayolas. Make sure every drawing has an appropriate title. Usually it's whatever you're looking at. And then that's going to be at the top, and then below the drawing you're going to put the total magnification. So if you're drawing it on low power, the total magnification is 100, and if you're using it on high power, the total magnification is 400. Okay, another couple rules that I have, all label lines must be drawn with a ruler. Okay, the second thing is you must label to the right of your drawing. So I'm going to show you on the first one how to do that, how to construct it appropriately so that you get a good grade. The next thing that you see, these are my rules for how to focus the microscope. I'm going to walk you through that. And then down here, these are the different specimens that we're going to be looking at today. So the first one, it's underneath the title Eukaryotes. So we're going to look at plant cells as well as animal cells. So the first thing is to look at something called an Acura plant. It's an aquarium plant. Okay, so we're ready to prepare what we call a wet mount slide. So I want you to see the setup that I have in the classroom. This is what it looks like at the lab table. I've got several things. You can see I have microscope slides. That's what these are. In the little petri dish, I have cover slips. They're little squares. You can kind of see them like that we put on top of the slide. I have chemicals. We're going to use iodine for this lab. We're going to use a little bit of water. And some specimens will use something called methylene blue. Okay, I've got toothpicks for the cheek cell aspect, and then this is the Anacaris, which is the aquarium plant. I'm also going to have a little bit of onion, and then we'll have pond water as well. Okay, so today we're going to start with the Anacaris plant first. So there it is. I have pulled off a single leaf. You can see it on my finger, and we're going to place that according to the instructions, place it on the microscope slide, and then put a drop of water on top of it. Okay, so I'm going to put that on there. We're going to take our water and just add 1 drop of water to the top of that. Okay, you can see that went on. And then we're going to cover it using a cover slip for the microscope slide. So it's just a square slide. I'm going to take it at a 90-degree angle, and then I'm going to drop it down. Okay, look closely as I drop it. You'll see the water will dissipate, kind of spread out. Okay, so that's what we call a wet mount. A wet mount is anytime you prepare something on a slide with a fluid, a liquid, and then you put a cover slip on top of that. All right, so I'm ready to examine the Acura plant on the microscope. So the first thing, these microscopes have mechanical stages. Do you see this little knob? So I'm gonna pull that knob out and then place my slide on the stage and then let that arm come back and hold my slide in place. Now I have to make sure I'm on low power, that's the 10, so I'm gonna click that into place, you can hear it click. I'm gonna come on this side because I wanna show you guys how cool these are. These microscopes have mechanical stage knobs that move this back and forth and up and down. So I wanna make sure I don't want it off, I want it to be centered, so I'm gonna move it kind of like that, but then I need to move it so that's right underneath my light. So you see how I'm moving that right and left? Okay, so now I have it centered. I'm gonna use the on-off switch, turn that on, and now I can see the light coming through my specimen. I just wanna make sure it's centered. There you go, that's pretty good. And now is the fun part. Too bad you guys can't do this. We're gonna use the coarse adjustment knob. I'm gonna look through the eyepiece until this comes into focus. So I'm gonna raise it until I can see whatever it is I'm supposed to be looking at. Okay, so I have the Acura focused on low power. I'm gonna see if I can look through this field of view with y'all. This is the hardest part ever. Okay, look what I see. Can you guys see that? You may wanna push the video so you can see it pause without me moving this thing all back and forth. See that? They look like a bunch of bricks everywhere. And that's low power, but you really can't see things very well. So what I'm gonna do now, watch the way that I do this. This is the important part. I do not wanna touch my focus knobs because I have it perfectly focused on low power. What I'm gonna do is just take this and click the high power in, and it is really close. Watch how close this is. It almost looks like it's gonna hit, but if I have it focused on low power, it will not hit the cover slip, and there it is. I have it focused. I have it clicked in. So that's on 40, so I'm now at 400 times. So I'm gonna look through the eyepiece, but as I do, I'm gonna fine tune it with this fine adjustment knob because it may not be perfectly clear. Okay, I have it focused on high power. What I want you guys to do, I'm gonna try to put it through on my phone. What you really need to do, because you're gonna draw your pictures from what you see, once you get to this point in my video, push pause so that you can see this for an extended amount of time because you're gonna need to draw one cell. Okay, so I'm gonna see if I can do this. Here we go, here we go. This is the hard part. Okay, that's where I would push pause right there. Push pause. That's actually pretty good. I got that for a long time. So you notice when you look through here, you actually see the dark black lines, but look where there's a black pointer inside the microscope. It's in the bottom left. Do you see that black line that comes out? Look at the tip of that. I have that pointing to one cell. Okay, do you see that? So there's one cell that is at the end of that pointer at the point where it looks like an arrow. That is one plant cell. So you need to draw one of those on your page, but put the detail in and then color what you see. Okay, so I found an image I wanted to show you off the computer so that I could point to some things for you guys so you would know the reference. So underneath the microscope, you saw these dark lines like this. This is actually because you're looking at 2 layers of cells. So I want you to draw only one cell. For example, look at my pencil. This is the cell wall right here. If you go around, keep looking where I'm pointing right here. That is one plant cell right there. It looks like one brick. So the next one will be right next to it right here. This would be the second one. Like that. Don't pay attention to those dark black lines that you see. You want to pay attention to the smaller ones. So this would be the cell wall right there going down around, and then all those little green dots are the chloroplasts. So you're going to draw one cell on your answer sheet like this, but make sure, because you're going to put your title up here. You're going to make a descriptive title of what you're looking at. Put your total magnification at the bottom. So it's going to look something like this. Put your total magnification. But your picture, I'll just kind of sketch one for you. Let's say we do this. Set your picture off to the left. because you want to leave enough room to the right so that you can label using a ruler. Okay, now I don't have a ruler, but I want to make sure you do. So you would bring your line out to the right like this and then label this cell wall like that, because that's the cell wall. Okay, then you would draw a little bit more detail because you can kind of see the cell walls thick like this. So I'm doing some detail. And then I'm going to put my little chloroplast in here. You want to try to make it look as neat as possible as what your picture is. Now, I'm not a very good artist, okay, but you can see that. But then as far as what do you label, this is where you go back to the lab sheet right here. It says right here in number 5, label the following structures: cell wall, cytosol, cell membrane, chloroplast. So you have to use your notes so you know what these things are, but obviously the cell wall is the outside. But my next one, let's say I want to label the cell membrane. I'm going to come just below this label, and my cell membrane is right there. Use a ruler, bring it out, but I'm going to line my labels up. This is an imaginary line right here. Don't do what I'm doing, but that's an imaginary line. And then I would put my label for cell membrane. Do you see how I have my labels nice and neat lined up? And if you do a ruler and do that, it makes a really nice scientific drawing. So the last thing is you'd grab some colored pencils, shade it the color that you see it, and then your first drawing is done. So we're going to continue this for the next specimen. If we flip this over, we're now going to look at an onion cell. Okay, and then we'll look at some animal cells. We'll do cheek cells, we'll look at pond water, we'll look at some other things on here. So that's your first specimen, and then we'll get ready next and do the onion. Okay, so we're ready for our second specimen. This is going to be the onion. So I have my lab sheet, and we're looking at this set of directions. It says obtain a piece of leaf from an onion bulb. So I have a piece of a white onion, you can see right here. What we want to do is remove a piece of the epidermis. So this is the tricky part. On a scale, like see this onion, on the inside, see how it's shiny? There's actually a thin layer of skin on the inside of the onion. So I'm going to use my fingernail to peel off a little bit, and then I'll show you. All right, here's the thin layer of skin. See that? So I'm going to peel that off and then place that on my microscope slide. All right, so according to the instructions, it says on step 3, place a drop of iodine stain on the slide and then place the onion membrane on the slide, taking care not to wrinkle it, and then put a cover slip on. So this is a cool thing to tell you guys. The reason we're using iodine is iodine tests anything that's a polysaccharide. It should stain it like a purplish black. It doesn't have to be dark black, but it'll stain polysaccharides like we did in the carbohydrate lab. Well, look what I'm holding. I have onion. Onion's a plant. Plants have cell walls made of cellulose. Cellulose is a polysaccharide, so that should stain the cell wall and make it visible underneath the microscope slide. So here's my drop of iodine. I have it ready to go on my microscope slide, and I'm going to take the onion and drop it in the iodine. This is the hard part. Okay, now it's wrinkled, so I'm going to flatten it out. So the onion slice, the epidermis is flat. So we now want to take and put the cover slip on top of it to protect our microscope. So now we're ready to go. We're going to repeat the process, but look what I'm going to do to my microscope. I have to put it back on low power, that's 10x, put the stage all the way down, and I'm going to start all over again, just like I did with the an Acura plant. So I'm going to put that in, there we go, let the arm click it in place. It's on my low power. I'm going to make sure it's centered using my adjustment knobs. That looks pretty good. And I'm going to try to focus it on low power. So this worked beautifully. Check this out. This is the onion on low power. Wait till you see this. It looks really good. You might want to pause the video when you get to a good shot of it. There it is. So you'll notice the yellow, kind of the yellowish color, that has stained every nucleus inside the onion cells. It's also stained the cell wall. That's so cool. Okay, that's low power, so don't make your drawing on that. We're going to switch to high power now. So now that we're good, I'm going to switch this to 40. It's really close, but it's okay. I'm going to use only the fine adjustment knob and see if I can get it a little bit clearer. Okay, so I have it on high power and it's ready to go. So as soon as I get it focused in here, remember push pause. There we go. Check out that specimen. If you push pause, you'll be able to see that yellow dot at the end of the pointer. See my pointer, the black line? That's the nucleus. And that's one cell that you can see, and then of course there's cells above and below and beside. But that is a pretty good specimen, and I'm impressed that I was able to get it like this long. That's what you're gonna draw, and then you